Bismillahirrahmanirrahim. Assalamu alaikum friends. In this video tutorial we learn about Watson and Crick model, which is very important model about the structure of DNA molecule, and he proposed the structure of DNA. So after the X-ray crystallography, Watson and Crick proposed a model which is basically called Watson and Crick model, and he got Nobel Prize in 1953. Now coming to explain the main point of Watson and Crick. So the first point of Watson and Crick is that DNA is consist of full immersed chain of nucleotide. So what is mean by polymer? We know that when small molecules are combined with each other, or when monomer unit are combined with each other, they form polymer. So nucleotide is basically monomer unit. When they are combined with each other, they formed a large polymer molecule, which is basically called DNA, deoxyribonucleic acid. So the first point of Watson is that basically DNA is consists of polymers chain of nucleotide. This is the structure, the one structure of nucleotide. This structure is basically nucleotide. When these nucleotide are combined with each other, when these monomer unit are combined with each other, they form polymer. If you learn one structure of nucleotide, so you can easily understand the overall structure of DNA. So now coming to the second point, two nucleotides are attached with each other by the help of phosphodiester linkage. So this is I draw here the structure of two nucleotides. So these two nucleotides are attached with the help of phosphodiester linkage. This is the phosphodiester linkage. Why we call this bond or this linkage is phosphodiester? Because this linkage are found between two nucleotides, is we call the phosphodiester linkage. So Watson and Crick say that two nucleotides are attached with the help of phosphodiester linkage. Now coming to the third main point of Watson and Crick model. So in 5 carbon of pentose sugar provide phosphate, and 3 carbon of pentose sugar provide hydroxyl group for this linkage. Now see in this place the third number carbon, I draw the structure of phosphodiester linkage, which these two nucleotides are make this structure. So here I draw the structure of one nucleotide. So this is the structure of nucleotide, and first number carbon nitrogenous base are present, and second hydrogen and second carbon. This corner show the number of or the position of carbon. This is called corner. So in second carbon, the hydrogen and another hydrogen atom are present in DNA molecule, and the third carbon hydroxyl and hydrogen are present, and the fourth carbon hydrogen and another carbon are present, and this and H3PO4 or phosphoric acid are present in 5 carbon. So here Watson say that in 5 carbon of pentose sugar provide phosphate, and this and for the formation of phosphodiester bond, the 5 carbon, this is the 5 carbon of one nucleotide molecule. So here the H3PO4 and this phosphate group are provided by 5 carbon for the formation of phosphodiester linkage, and Another is 3 carbon of pentose sugar provide hydroxyl group for the formation of phosphodiester bond. So this is the third number carbon which provide hydroxyl group for phosphodiester linkage formation. Now coming to the fourth main point, these polynucleotide chain warp around helically to each other. So I draw the structure of overall DNA molecule. So here the space are very less. This is one strand, this is another strand. So he said that these polynucleotide mean what? This is a one polynucleotide chain which is 5 and 3, and the another is 3 polynucleotide chain. They are helically around with each other. Now coming to the pep point, which is basically sugar phosphate on the outside and purine and pyrimidine are the inside of the helix. So what is purine and pyrimidine? We know that adenine and guanine are called purine and cytosine and thymine and uracil are called pyrimidine. So purine are basically double ring molecule and pyrimidine are single ring molecule. So he says that sugar phosphate on the outside and purine and pyrimidine are the inside of the helix. So here the purine and pyrimidine, we know that in this side the adenine and thymine are attached, so these are inside of this polynucleotide chain, and the sugar are outside and phosphate are also outside from this two strand of nucleotide. Now coming to the sixth number point, the two strands are complementary to each other, mean that this is the one strand of molecule of DNA molecule, and this is the another strand. We know that DNA is found in two polynucleotide strand structure. So this is the Watson say that the two strands are complementary to each other. So suppose that is adenine must match with thymine, cytosine match with guanine, T, C, G, A, T, A, T. Say that mismatching are not occur in these strand. So the 7th number point of Watson and Crick that 10 nucleotide occur in per turn. What is mean by per turn? So here's from this region to this region, this is called per turn, and this per turn 10 nucleotide mean that 10 monomer unit are found in this place or this per turn. So this is the 7th number point of Watson and Crick. The distance between two nucleotides are 3.4 angstrom. So the mean that this is the one nucleotide and this is the another nucleotide. So the adjacent distance distance between two nucleotides are 3.4 angstrom. The distance between two nucleotides are 3.4 angstrom. Simple way. Now coming to the 9.2, major grooves are found in the structure of DNA. One is called major grooves and another is called minor groove. So here see in this place, this is called minor groove, and this is called major grooves. Now coming to 10 point, that is basically adenine and thymine match with double hydrogen bonding, and cytosine and guanine match with triple hydrogen bonding. So this is the Watson and Crick model about the structure of DNA. Hopefully you learn this structure.